The detail provided in this protocol addresses human skin equivalent generation, which is often technically challenging. Novel human skin equivalent protocol elements such as vasculature and volumetric imaging are included.
The technique enables the straightforward construction of a skin model that more closely matches in vivo tissue for the study of diseases, aging, and personalized medicine.
The most challenging part of this protocol is the transition between submersion and air-liquid interface. It will likely take a few trials to optimize the timing and media levels for consistent results.
To begin, add 516 microliters of media to a cold cap tube and immediately add 375 microliters of cold collagen using the 1000 microliter positive displacement pipette. Remove the empty pipette tip and switch to the prepared 250 microliter positive displacement pipette to mix. Mix quickly but gently to prevent excess bubble formation, keeping the tip in the solution. Mix uniformly from different positions of the tube until the solution is of homogeneous color, which typically takes about 5 pipette cycles or 10 seconds. Immediately disperse 125 microliters of acellular collagen onto the membrane of the 12-well culture insert. To ensure uniform coverage, tilt the plate and use the pipette tip to paint the membrane by gently spreading collagen around. Immediately move the 12-well plate to a 37 degrees Celsius cell culture incubator to let it gel for at least 20 minutes. After the gelation period, take the 12-well plate of acellular collagen out of the incubator. Remove the 1.7 milliliter capped tube from wet ice, then remove the stock collagen.